The history of molecular biology can be essentially begun with the idea of from whence did heredity come? Was it originally an ethos? Was it a spirit? Or did it have a molecular basis? And for quite a long time, many biologists honestly thought that heredity originated in proteins. And several experiments, mainly in the United States and the United Kingdom, actually show that nucleic acids and not proteins were actually the source of heredity. All of this is key to understanding today's recombinant DNA technology and all of the revolutions that are involved in modern-day genetics. We can honestly begin at the Rockefeller Institute in New York City with Oswald Avery and Chargaff. Oswald Avery was the first to actually show that DNA, or nucleic acid, heretofore believed to just be an extraneous biosynthetic byproduct of the body's physiology, was actually responsible for DNA. Avery's original experiment showed that if one were to take DNA out of a frog egg, remove its DNA completely through chemical means, and then allow the frog egg to generate or germinate, it couldn't form a frog. Whereas if the proteins in its nucleus, the center of the cell, wherein all of purportedly the hereditary information was stored, were removed, but the nucleic acids remained, the frog could develop normally. Although Avery did the crucial experiment that showed that DNA was, of course, the source of heredity for all living things, he never won the Nobel Prize. Chargaff was an Eastern European scientist who came to the Rockefeller Institute in New York as well, and showed that DNA in and of itself had all of the characteristics that are actually needed for heredity. It provided information at a level of very low entropy, that is very low disorder. It could show through repeating patterns that there was enough variability in the sequence of DNA and the chemicals that are contained within DNA to provide information for even extremely complex living organisms, from very basic yeast to human beings that have the ability to think. A very exciting idea indeed. But no one could really honestly think of the mechanisms of heredity until the Englishman Francis Crick and the American James Dewey Watson actually discovered the structure of DNA, not through any direct means, not through X-ray crystallography, not through chemical identification, or any of the modern-day techniques that were used, or even the techniques that were contemporary to them, developed by Bragg and Rosalind Franklin, but instead used very simple stick and ball models to come up with the idea that DNA could, first of all, by separating its two strands, opening up to the cellular environment, provide the information that was necessary for the machinery within living things to take its information and turn it to the vital structural proteins and vital enzymes that organisms need to just survive. The next step, once we have the idea that DNA has a given structure and has information inherent within its structure, that is, it can give information to the cell through a functional means, we have to understand how we can manipulate it. This revolution came back actually in the 1970s with Daniel Nathans and his group at the Johns Hopkins University, who discovered that bacteria have very interesting enzymes that allow them to destroy the DNA of viruses that infect them. These enzymes, referred to as restriction enzymes, allow the bacterium to go and chew up enemy DNA. Luckily, molecular biologists realized that these very enzymes can allow them to manipulate the DNA in very interesting ways. Because of the nature of the enzyme, it will look at DNA, specific codes within DNA, and clip only those specific codes, such that one can now cut and paste, so to speak, using the lingo of modern-day Macintosh or personal computer technology, could cut and paste DNA and manipulate it, such that one could get the information from the DNA that one actually wants. This discovery of restriction enzymes, along with the discovery of the structure of DNA, these two discoveries were actually awarded the Nobel Prize. The importance of restriction enzymes and the importance of research that has been done since the 1970s is crucial to recombinant DNA technology, such as the idea that DNA that has been manipulated in the test tube through the use of restriction enzymes can now be introduced into cells, and that these cells can therefore change their entire physiology just on the basis of a simple man-made manipulation of their genetic information, has essentially caused the massive explosion of biotechnology this more important than anything else. Of all of the aspects of the revolution of molecular biology, the economic aspect has been of great interest to industry, and therefore has caused a massive influx of funds into molecular biology and has introduced further innovation.